Hello, Dr. Papa Michael. This is Shoptaporni. I recorded this video an hour ago, but somehow it did not work out. So I'd be recording again. This is the second assignment that you gave us to do. The first question asks us to find the number of mutations and indels that the sapien vaccines have from their corresponding wild-type versions of the poliovirus. The way I found out this was I went to NCBI and typed human poliovirus, and I searched it. In entrance we would get the nucleotide sequence, which is 5335. There are other sequences, protein sequences, conserved protein domain sequences, unigene, but I would like to go to the taxonomy part because that would give me a single sequence and I can play with it. So let's go there. Here I find the human enterovirus C. I'm clicking that. From here I can find out that there are 3 strains of poliovirus. One is human poliovirus strain 1, poliovirus strain 2, and poliovirus strain 3. And the vaccine is made of, the sapien vaccine is made from poliovirus strain 1, poliovirus strain 2, and poliovirus strain 3. So let me click the poliovirus strain 1 and I click the human poliovirus 1 Mahone, which would give me the nucleotide sequences of the poliovirus strain 1. I click that and it takes me to human poliovirus 1 Mahone complete genome, which is 7440 base pairs. I click on that and it gives me the GenBank format of the human poliovirus 1 Mahone complete genome. From here, from the right side of the page, I can see that there is a tab called Analyze this sequence, under which we have Run Blast. I would run the blast, click, there is the accession number comes here, and I'd keep rest of the parameters as it is. This is a nucleotide collection NRNT, so I'd keep the rest of the parameters same and then blast the sequence against the database, and let's find out what we get. This takes a little bit of time. So we have got our match from the database, and the red color usually indicates that there is a good match. Let's go to the sequences and then find out our virus that we were searching for. Human poliovirus 1 Mahone complete genome. And from here we can also find that there is another virus strain, which is called the sapien virus 1, which is the vaccine for poliovirus, which is also called oral poliovirus, and it is made up of 3 strains, but mainly this is the strain made of strain 1 and its complete genome. If we click here, it would take us to the GenBank format of the viral strain. We are going to click the max score, so it gives us the alignment. Here we can find the score, expect value, identities, gaps, strand, which kind of strand it is, and then the sequence alignment with our query sequence. So that is what we get. and this is how I found out the rest of the other two, which was human poliovirus 2 and human poliovirus strain 3 and their corresponding vaccines, and found out the matches. In the second question, you've asked to using BLAST, using BLAST, what is the probability that the sequences with accession number this and this are unrelated. So here we have to go to BLAST again, www.ncbi. Okay, from here I'm going to BLAST. This is also a nucleotide BLAST. So here I'll put the accession number, and here there is another thing, which is there's a checkbox which says align two or more sequences. We'd click that and we'd get another place to add our sequence, the second sequence accession number. We copy paste this, and then paste it here, Control V. Okay, so now we have other parameters to look into. I've already tried searching with highly similar sequences with the Mega BLAST, but it does not give any results. So we would try with somewhat similar sequences, which is BLASTN, and let's find out what the alignment would be like. So I BLAST it and found out that there is some similarity between the two sequences, because it reads as Streptococcus species, which is this is a gene for 16S RNA partial sequence, and this is uncultured archaean gene also for 16S RNA, and we all know that the 16S RNA are conserved across species, so there may be some differences, but there has to be some similarities as well. So there is a dot matrix view which would tell us how similar the sequence is or how dissimilar the sequence is, and from the E values we can find out that it is not close to 0. So the E values are high, which means the matches may not be as significant, but there is 72% of identity. Maximum score is 228. So we found out the alignment of the sequences, alignment of the two sequences, and found that there are gaps in between. So so here you have asked about the E-value score. For a high scoring pair, the E-value should be negligible or as close to 0 as possible, so that when the E-value is very low, it means there is a significant match, and it means that that sequence would not arise randomly, and it also means that it's a statistical parameter, and it says e-value specifies a statistical significance threshold for reporting matches against database sequences. The default value 10 means that 10 such matches are expected to be found merely by chance, according to the stochastic model of Carlin and Achtul. If the statistical significance ascribed to a match is greater than the expect threshold, the match will not be reported. Lower expect thresholds are more stringent, leading to fewer chance matches being reported. So the higher the e-values, essentially the e-value describes random background noise. The lower the e-value, or the closer it is to 0, the more significant the match is. So this is what I tried to cover. And is this a probability? Yes, it is a probability. And in the third portion of the third question, you have asked that to post 2 links or 2 blocks on the Blackboard, but there is no tool to add. There isn't any new tool to add a blog, but I did add 2 URLs to the wiki page. One is for ZFIN and another is for Wormbase. One is for zebrafish and one is for Sonoroditis elegans, and another I added for RNA regulatory regions. So regulatory RNA sequence database. And the fourth question asks us to find a protein, a hypothetical protein, and then search against another protein sequence so that we can find the similarity between the two using 2 different matrices, substitution matrices like PAM or BLOSSOM. And how I found this out was I went to XPSI, which is a part of Swiss Institute of Bioinformatics, and I found out random sequence, which RANDSEQ, which gives the random protein sequence generator. Sequence would be around 200 amino acids, and I just submitted to generate a sequence, which gave me a sequence which was, this is a random sequence, so it is not going to match with mine. The first time I did the sequence was different from this one, so I would take the sequence that I found out and copy it and I'd go to BLAST again. Okay, so I'll go to BLAST. Here I'll go for protein BLAST because this is a protein sequence. Here it pastes the protein sequence. From this protein sequence I've made some changes and made another protein sequence of my interest, and this will be my second sequence with which I would want to align the previous sequence and see how much matches I find with the two substitution matrices. So I'd click this one, align 2 or more sequences, and I would paste the small sequence that I randomly did. And here for algorithm parameters I would go for BLOSSOM 62 or BLOSSOM 80 or 45 because I've already searched with PAM and I saw that the below some 80, and 45 gives me a good score. So